Chlorotic streaks an important disease in our industry. It occurs in all districts and it has a very long history. We've known about it since the late 1920s. And it's characterised by these long, creamy, white streaks on the leaves. And we know that it's spread through the roots, and it's spread in wet soil and floodwater and irrigation water. It reduces the yield, and it also reduces germination and regrowth of ratoon crops, so it is quite a severe disease. Chlorotic streak is quite famous because it actually went about 80 years without anybody in the world knowing what caused it. But our group, which was based here at Indooroopilly and at Woodford, we managed to sort out what the causal organism was. And it's a thing called a cercozoa, which is a bit like a protist or an amoeba, and that is a very simple-celled organism that swims around.
And so this has led to a new project. What are you trying to do? What's the new project about?
Well, finding the cause was extremely exciting, but that in itself is not really useful. What is more useful is the fact that we can now use the organism grown in pure culture, and it means we now have control over the disease. So we can now control the way that we inoculate plants. So we can use that to actually develop a screening procedure to try and work out the resistance ratings of our varieties.
And that's quite a practical outcome, isn't it? Yeah.
It is, because whether or not plants are resistant and what their ratings are for chlorotic streak is very confusing, and we've had a lot of trouble getting reliable results. So we're hoping that we can develop a faster, sort of high throughput screening procedure.
And so this is culture here, is that what we're looking at? Or?
So we found that the organism actually lives in a really high concentration in the stalk of sugarcane. So we were able to take small pieces of the stalk, and in pure culture we use these little flasks, and we use a media that's actually made out of potatoes, and the organism comes out of the stalk and lives in the liquid media, and it swims around. So that's an early one where you can still see the pieces of stalk, and then once it gets going we sub it and we start growing it in these much larger flasks.
And you use that to inoculate?
And this is what we use to inoculate the sugarcane plants.